All right, so we want to start off by going over the cell cycle. So we're going to kick things off by studying the cell cycle. The cell cycle is an orderly set of stages from the first division of a eukaryotic cell to the time the resulting daughters divide. Just before the cell begins to divide, it is going to undergo a series of transitions. So first, it's going to grow much, much larger. The number of organelles are going to double, and the cells are going to undergo DNA replication, which is going to lead to the doubling of the genetic component. So that first stage of growing and doubling is going to be the first of two major stages of the cell cycle. It's known as interphase. So that interphase is going to be the cell carrying out its normal stages and then growing and getting ready to divide that genetic material. The second stage is called the mitotic stage, which is going to contain two phases, mitosis and cytokinesis. Most of the cell cycle is going to be spent in that first stage of interphase. This is when the cell is going to carry out all of its usual functions. So if it's a muscle cell, it's going to be producing massive amounts of ATP. If it's a liver cell, it's going to be carrying out detoxification. If it's a pancreatic cell, it's going to be producing insulin. During interphase, the cell is doing whatever it is that cell is supposed to do, but it is also going to be going and it's also going to be getting ready to divide. Like I said, it's going to grow, the cell itself is going to grow larger, the number of organelles is going to double, and the amount of chromatin is going to double as well. That DNA is going to undergo DNA replication, which is a very specific stage that we will talk about in a later chapter. But that first set, that interphase, is going to be divided into three main stages. So here are the different stages of interphase. In the G1 stage, like I said, the cell is going to be performing its normal function. However, it's going to also be doubling its organelles and getting the materials ready it needs, or getting together the materials it needs for DNA synthesis. DNA synthesis is also just known as DNA replication. The S stage is the DNA replication stage. The reason it's called the S stage is because this is the stage of DNA synthesis. So G1, the G stands for growth, the S stands for synthesis, and the G2 stage is the second phase of growth. So our first stage, growth phase, is when the cell is going to carry out its normal functions, double its organelles, and gather the materials it needs to undergo DNA synthesis. In the S stage, the DNA replication is going to occur, or DNA synthesis is going to occur. After the S stage, each chromosome is going to consist of those two identical sister chromatids. So if you remember when we talked about in the previous section that we have a single chromosome, once it replicates, it's going to form two sister chromatids that are attached together at the centromere. After we complete our DNA replication or our DNA synthesis, we're going to move on to the G2 stage. In the G2 stage, we're going to synthesize the proteins that are needed for cellular division. So we're going to continue to grow, but we're also going to gather up whatever we need to undergo cellular division. So here's a really great diagram that kind of shows what's happening in the stages of the cell cycle. The amount of time that the cell spends in interphase is going to vary widely. Some cells, like nerve and muscle cells, typically do not complete the cell cycle. They are permanently arrested in the G1 phase. Because they won't ever continue to the G2 phase, we instead say that they are in the G0 stage, meaning that instead of progressing, instead of duplicating, they're going to be kind of shuffled off into this area over here, which we call the G0 stage. Embryonic cells, on the other hand, spend very little time in the G1 phase. They can complete the cell cycle in only a few hours. In just a few hours, they can undergo growth, DNA replication, growth and preparation for cellular division, and then they'll actually go through that cellular division very, very rapidly. So following interphase is going to be the cellular division. Once we move out of interphase, we moved into the mitotic stage, or also known as cellular division. Cellular division can be divided into two stages, M stage for mitosis, and then cytokinesis. The mitosis stage is going to be a specific type of nuclear division. Like we said, mitosis is going to be responsible for growth and repair. Cytokinesis or mitosis, the stage, the cell cycle division as a whole, is going to be used for growth and repair. So mitosis is simply a specific type of nuclear division. It's going to be followed by cytokinesis. Cytokinesis is going to be division of the cytoplasm. The cell cycle is going to occur continuously in specific tissues. Your skin cells are constantly growing and repairing and sloughing off and dying and being replaced again. So in certain tissues, we're going to see a constant stage of mitosis occurring. Mitosis is going to be balanced by the process of apoptosis. Apoptosis is programmed cell death, and it occurs when cells are no longer needed or have become so excessively damaged that they cannot continue on. There are a couple of mechanisms that we can use to control the cell cycle. The cell cycle is going to be controlled by checkpoints. Checkpoints are going to delay the cell cycle until certain conditions are met. So we have a G1 checkpoint, G2 checkpoint, and an M checkpoint, the mitotic checkpoint. The cell cycle could also be controlled by external factors, things like hormones and growth factors. Failure of the cell cycle control mechanisms can result in unrestricted cell growth or cancer. This is why the cell cycle checkpoints are so very, very important. They prevent cells from undergoing rapid and uncontrollable growth that can lead to the development of cancers. The G1 checkpoint, that's going to be this first stage right here. The G1 checkpoint is going to occur if the cell cycle passes this checkpoint. The cell is going to be committed to divide. If the cell does not pass this checkpoint, it can enter that G0 phase that we talked about, where it's going to perform all of its normal functions, but it's not going to divide and replicate. This is going to ultimately be controlled by proper growth signals, like growth factors. These must be present for a cell to pass the G1 checkpoint. The integrity of the DNA is also going to be checked at this phase. If the DNA is damaged, proteins like p53 can actually stop the cycle at this checkpoint and place the cell in the G0 phase. So if that DNA is damaged, the cells can carry out their normal functions, but they're not going to replicate that damaged DNA in the S phase, and they're certainly not going to carry that out into future generations. If the DNA can be repaired, it may re-enter that cell cycle. So we might, while we're in our G0 phase, might repair that DNA if we are able to. But if we're not able to repair that DNA, the cell might undergo apoptosis. At the G2 checkpoint, the cell halts here until the cell verifies that the DNA has replicated properly. This is going to prevent the initiation of the M stage unless the chromosomes are properly duplicated. If the DNA was damaged during that replication process, arresting the cell cycle allows time for the damage to be repaired so that it cannot pass on that damaged or that mutated DNA to daughter cells. So we're going to stop here. We're going to repair that DNA if we can. If we cannot repair that damage, apoptosis will occur. If we are able to pass that G2 checkpoint, we're going to move on to the M stage. We are going to right away undergo mitosis. We'll begin the prophase, prometaphase, and metaphase stage. However, there is another checkpoint right at this stage that is going to occur between metaphase and anaphase. This is going to make sure that the chromosomes are properly attached to spindle fibers so they can be distributed accurately to the daughter cells. I like to call this as the seatbelt check. And you'll see why a little bit later on when we talk about metaphase and anaphase. All you really need to know is that at this stage, we're going to make sure that we are ready to carry out our nuclear division. Here, prophase, prometaphase, metaphase, this is all gearing up. This is like loading everybody up into the roller coaster. The seatbelt comes down, and then we've got our workers on either side of the roller coaster running down the line checking seatbelts. Once we check that the seatbelts are in, we can proceed with the rest of the ride. But you'll see all of that a little bit later on. I just want to point out that's really what the M checkpoint is. It is a seatbelt check to make sure that everything is ready to go before we proceed with the rest of the mitosis. There are a couple of external controls that control that cell cycle. an external signal like a hormone or a growth factor can stimulate the cell to divide. It's going to bind to a receptor on the plasma membrane of a target cell. The signal is then relayed from the receptor to the proteins inside the cell. The proteins form a pathway called a signal transduction pathway. This is where this cell is going to, or this protein is going to activate this protein, which is going to activate this protein, which will activate this protein. This is going to pass that signal down from one protein to the next. The last signal of the signal transduction pathway is going to activate the genes in the nucleus. The expression of those genes could stimulate or inhibit the cell cycle depending on the type of gene that it is. Genes that are called proto-oncogenes are going to stimulate the cell cycle. Genes called the tumor suppressor genes are going to inhibit the cell cycle. We're going to talk about proto-oncogenes and tumor suppressor genes quite a bit in our next chapter when we talk about the development and formation of cancer cells. All right, so you should be able to describe the cell cycle and list the locations of each phase and their checkpoints. You should also explain the purpose or explain the specific purpose of the S phase of the interphase cell cycle. Explain how the checkpoints help protect the cell against unregulated cell growth, and summarize why the external controls might be necessary to regulate the cell cycle. We haven't talked extensively about the external controls, so if you would like to, I would recommend that you read this section of your textbook and hold on to that information. This will come in very handy when we talk about cancer, the development of cancer, but it's not going to be necessary for any homework or for the homework or the quizzes associated with this particular section and chapter. So review this, read over it, and then keep that in your back pocket for when we talk about it in our next chapter. All right, that'll be it for this section. Please let me know if you have any questions, comments, or concerns.